Okay, so we will start the session now. Okay, before starting the session, we'll have some interaction. If you people are okay with that. So actually, this course is for that statistic for biomedical engineers. Like I'll give the brief about myself also. So can I have some some sort of about that is yourself? Can someone please tell about yourself, from where you are, that which course you are studying?
Mm, If is it okay? I go first. Yeah.
Please, sorry.
Can I go first?
Yeah, yeah, yeah.
So, hello, sir. Myself, Meg Mhatre. I'm a graduate student. I've completed my graduation in biomedical engineering, and I have joined this course to understand statistics with regards to doing research and do means to draw conclusions for signal processing and image processing techniques.
Okay,
Thank
so
you.
that you are doing the PG now, like MTech.
No, sir. I'm on a gap year, sir.
Okay, okay. Uh, can someone the next, can you uh, introduce yourself for a while, then so that we can start the session once the that some people will join. Anyone? Yeah, yeah. Okay. Mm, Lokant Reddy.
Hello, sir. Myself, I'm Loknatha. I have done my master's in clinical engineering from IIT Madras, and right now I'm pursuing my PhD. So I thought this course would be really helpful for me to work on my PhD work. So that's why I have taken up this course.
Okay, fantastic, fantastic. Uh, can someone that next? Just give the brief, like what is motivation and that what you're doing.
Good evening, sir. I'm Devashree from Loyola Academy Degree and PG College. Now I'm pursuing first year in from BSc Biotechnology and Chemistry Genetics. I'm interested in this course so that I'll gain experience from biomedical engineering, in especially in statistics. So I joined this course. Thank you, sir.
Thank you. Can someone last, then after we'll say start the session. Or shall I go ahead? And we'll, because this first week is full of that why do we need to study and full that basics we'll be going to see. Shall I start the session?
Yes, sir.
Okay, so before starting that, that my name is MD Aziz Raza. I am PMRF scholar in the Department of Mechanical Engineering, Nagore Petrapalli. And this course is on the statistics for biomedical engineering, and the course instructor is Dr. Babaji Srinivasan. And this course is going to span for 12 weeks. So we are going to start the fundamental of the statistic 2, that we'll see the design of experiment also, and we'll see full that from the description statistic to the inferential statistic we'll see it. Just a minute, I'll start my recording. Okay. So before starting the session, so what I'll do, I'll give some brief. about the myself. Like I am doing the like PhD in the field of sports biomechanics, and I am the PMRF fellow from the July 2023. And my that research area is actually sports biomechanics, and we'll deal on the muscles physiology and sports performance analysis. For that, we'll use that EMG analysis. Before that, I was in the mechanical engineering that I have done machine design and all. And as part of the PMRF, that we have to take some TA courses outside. So that I took the last time biostatistic and design of experiment, and some lecture that outside of the college also I took. And at NIT Trichy also we have to take some internal courses, like I took the graphics quality control that deal with the statistic and all, and the basis of mechanical I also took. So this is just a brief about me. So now we'll come to the statistic. Okay. If anyone have the doubt or something, please just ask the question. There is no issue with that. Okay. So once we'll talk about the how the course is going to span out. Okay. So first week, uh, that you might have seen that it's full of introduction. We are talking about why do we need the statistic, and what is the statistic, why that is going to help you out in the future. Then it's coming to the biostatistics, like once it's go to the some specific area like biomedical maybe, so that become biostatistics. If we go to some sort of that is the industry, so that become the quality control, that aspect will come into the picture. Then we'll see their application, that how come it's going to help you out. Like say if any that drug or medicine that have been made, so that based upon their descriptive, that we can't come up with the conclusion like what is the efficacy of that particular drugs, or maybe some treatments are there that we are going to follow it for the any disease. So whether we'll say okay, in terms of description we'll say okay, that will go for the 6 month or 7 month or 2 week or 6 week. But what is the efficacy, whether it's going to help them out or not, so that will tell based upon that is the population or maybe the sample size. Then we are going to apply the statistic. So based upon the description or descriptive, we are going to study the inference about the that study. Then we'll discuss some few cases. So that is going all about the first week. Then after second week is going to be a like descriptive study. Descriptive means like it's a mean, median, mode. It's like something from there we can't conclude anything. Something like we can't say this that method is particular good or not, that we can't say based upon that. So for that we will see descriptive. To discuss all the statistic or descriptive statistic or inferential statistic, we need some sort of that is the role of the probability. Means we'll talk based upon the probability itself. So before starting, we'll in the third week we are going to that study about the some theory about the that is the that is the that is the probability. Then we'll check about the that is distribution. Then we'll check expectation, then population variance, and then we'll check. Then it after that it is going to start the inferential statistic. So that's going to fourth week. So in that we'll check the central limit theorem. In that we are going to whether the data is skewed or not. So that based something we'll check it out. After that we'll check the what is the confidence interval. Sometimes we'll say my confidence interval 99% chance is that particular device or drugs it's going to be a like efficacy. So for that, whatever the things we are going to make or inference, okay, so we have to formulate some hypothesis. So in that we'll check the what is the hypothesis, why do we need to do the hypothesis testing, what is that element of the hypothesis, what is the p value, what is what is the that is the chi-square value, what is the eta square value, and all we have to check it out. So it will going to span out types of the error. Once we talk the errors, so in general we'll say whether that the mistake, it we'll say the error. But in the statistic we have to tell different types of error is there. Type 1 error, type 2 error, based upon the whatever the hypothesis will make. So based upon the error will come. Then after we'll check the power of the test. Power of the test is again, it's like we are talking about the confidence. I will say 95% or maybe 99% or maybe the 90% based upon that our data sample data quality and all. Then will comes based upon the variance, what is the test, then contingency test and all. Then sixth week is going to be about when we have the different types of population. Okay, so then we'll check the t-test, then we'll going to discuss about the ANOVA. So further it will go, once we'll discuss about the inferential statistics, then we'll go to design of experiment. Once we are going to do the statistics, so for statistics we need the data. The data collection is very important in the statistics, because if we don't collect the data properly, what will happen? It's going to be like a data will be the very noisy, or maybe the data will be not correct, or maybe whatever the our objective that is not going to fulfill out. So that's why it's very important to have design of experiment very like a smooth way, or maybe the properly we have to design, so that we have a some sort of complete block design. That is that is the one more is there, blocking is there, then we have a square design, then factorial design is there. Then after, based upon their design, then we're going to two-way ANOVA and all, we are going to check it out. So this is how the course is going to expand. So this is first week. First week, if you have some basic doubt or something, so that we'll discuss it out, and then what is the use, where it's going to happen, then we'll discuss some few cases also. Okay, so we'll come with that. What is the need of a statistic in biomedical engineering? Can someone please tell? Like, this session will not go in one way. We will go like we'll have the interaction. We'll discuss and we'll try to get the better or the best of that. That is the best out of it. Okay. Can someone tell why do we need the statistic in biomedical? Like, we'll come to the specific. Can someone please tell? Why do we need statistic in biomedical engineering, or let's say that not in biomedical, in the general also, why do we need the statistic?
Excuse me, sir. Can I? Thank you for this opportunity to give this, sir.
Yeah, yeah, then go ahead.
Basically, the statistics in biomedical engineering, it helps mostly like designing experiments, like analyzing the data,
Okay, you are very close.
and that's it, I guess, sir.
Okay, uh,
Thank you.
any other?
Uh, if I may have a go,
Yeah,
I would
please.
say that for me, statistics is like to condense a very large amount of information into one or two values. Like if I'm having sets of like large sets of various data sets, so for each of the data set using statistic, we can just condense their overall information into one, means one value or multiple values as per our convenience or need.
Okay, that's so convincing. So a statistic is nothing as a data. Once we talk the data it going to contain the information, is it? Okay, when will data, or data is the number will give the some sort of the particular information. Okay, but the purpose of a statistic is to we are going to investigate or evaluate that what is the nature of the data, or maybe we'll going to conclude or will give that interpretation from the data. Let's say someone say this dose is that much number. Okay, this drug is that much number, so that is some sort of data. The data is giving some sort of information, but whether the data or that drug, how much efficacy is there, so that will going to talk based upon the statistic. Okay, it's something that we are going to interpret their efficacy, or maybe their what type of the nature of the data. So it's something we'll say, let's say someone is doing some sort of experiment they are doing. Let's say we have that heart rate monitor or the wearable EMG. So from there we'll get the some sort of data. So that data will contain some sort of information. Whether the data is reliable or it's going to be helpful or not, so that will going to based upon the statistic, is it? In terms of this is some sort of biomedical. And if we talk about any manufacturing plants and all, so that let's say some companies are making the bearings, ball bearing, or maybe some bangles or something. So whether we have to check their that quality, so we'll that we can't check for the all. Okay, so that number is, we'll take some samples, we're going to check it, and then we'll tell, okay, that quality check that have been passed. So that all things will tell based upon their based upon the statistic itself. So so that's why that is the statistic is something they are going to help in the quantify the variability. Okay, for a that what you can say that's a bearing and all, it's there, that is the life cycle. For a we have the BP monitor, so that will be blood pressure. So whether they're giving the accurate what is the their percentage changes is coming, or maybe if you're going to consider some data, so whether data is that normal or abnormal, so that we have to check. So they're going to, for that we have to check some threshold value, or maybe we'll have to check the that is the hypothesis. So it's something like we are going to tell about the data, and we are going to claim something whether the data is abnormal or normal, or that the particular devices is good or bad. Okay, so it's something we are saying that to quantify in terms of their efficacy, or that may be in terms of quality and all. Okay, so if we talk about the that is the statistic. So a statistic is something. Just 1 minute, like I'll be giving some brief of this full whole week, and then we'll solve some 7 to 8 questions based upon that. That will go to help for your particular quiz this week. Okay, so this is not a course actually. Whatever you will go and see that the lecture for that you can see in the week 1 content. Okay, so here I'm going to just conclude, or maybe we'll give the brief what's have been done in the that particular week. Okay, so that statistic, once we say statistic, so that is going to start from the collection of the data. Okay, let's say for a something sample and all something, so it will going to start from the collection of the data. Then we have to do some sort of fashion, or uh, organizing, or we have to present it. Then after analyze it, then we have to interpret. So it's going to give a more effective decision. Okay, so it's something they are going to give. So it's going to be like starting from the data collection, then it going the giving some sort of effective decision. So that full process will say statistic. Okay, so in that statistic where we can use in the medicine, public health, biological science, wherever there is uncertainty. Okay, whenever we have some certain things are there, so there is no problem, we can claim, we can directly say okay, this is good, this is bad. Whenever there is uncertainty, like let's say we have a different types of people are there, like let's say for the covid what happened, we have the different groups of the age, we have a different genders was there, and we were having some sort of vaccine came, but the variability was more, uncertainty was more, because some people having some that is the medical history, some was fit and young, some was having infant. So we have the uncertainty. For this type of study we need to study the statistic. Without statistic we can't come up with the some sort of any conclusion. So that's why, so in the biological sciences that's going to help you out. So that's why we'll say if it is that statistic, it's going to be a particular to the biology or biomedical, so we'll say biostatistic. Okay, so if we talk about some question, like some questions that will come when we talk about the uncertainty, like some drug that have that been described with the doctor, whether it's going to be a treat the illness or not. Let's say for a fever, we have different types of dolo is there, we have a paracetamol is there. So which one is more effective, or which one is giving more effective results and all, or maybe that certain genetic, that is the mutation we have or not, whether what are the chances in a population due to that effect, or due to that variability, or maybe due to that particular issue, what is the chances that she will, or that will get the breast cancer and all. Or if a some that pesticide for that will going to cause the cancer or not, that should be banned or not. So that all uncertainty that will be question is there. So that should be answered with the some sort of statistic, or maybe we have to formulate the hypothesis. Okay, to answer all this query we need, what is the need, we need the data. So for data, what we can do, data we can collect through the that is the routinely kept record. Let's say some pesticides have been poured in that some forms, and also they, that some person will going to record, okay, we have kept this much amount of pesticide in that form, okay, this is the their quantity, that is the name. So that will be going to 2 month, 3 month and all. So that will going to keep the record for a some 2 month, 3 month, 4 month. Either we'll do the survey, that we are going to float the form for a 4 months, or maybe those who are doing the particular field in that area will give the survey form, and then we'll collect the data. Or either we'll do the experiment and then external source. That is experiment, we are going to collect the data by ourselves. Or maybe some federal agencies are there, maybe that we have that ICMR is there, they're collecting data for particular things, that what is the COVID that happening, that every day, what was for the 1 week and all. So this is the data collection methods are there. So if we talk about, so experiment, so it's collected by the someone, those who are doing the work, or maybe the research, or the survey. Survey, like it's something, okay, some flu came, but we don't have the exact data, but we're going to put the survey. That some sort of we have in a region or area, we have 10 hospitals are there. So from the DM side, or someone that is someone authority, that they're going to put the form, okay, in your hospital or clinic, what is the number of patients that came, what was the severity, what was their that is the mortality rate, or some parameter they're going to check, and then we'll collect the survey. Based upon that, we'll say what was the severity of that particular flu and all. Or the same way, we have the records for the 6 month, or maybe 7 month, or 10 months and all. So if the same statistic and all that we're going to apply to the like physiology, agriculture, or maybe something, or maybe we're going to apply to the biology, so that will go to termed as a biostatistic and all. Okay. So if we talk about the biostatistic, that will go into in the field of healthcare, that will go in the field of healthcare, or maybe that is the environmental health, or maybe the medicine, or the biology and all. And importance of the role, and the role of the statistic is that we have to first, what is the objective? Based upon the objective, we have to study design, like we have to plan the design of experiment. Then after, we have a plenty number of, then we have the plenty number of the population will be there. Let's say population, can someone tell what is the difference between the population and sample size? Sample and population, can someone tell? uh, that you might have gone through that full week lecture. Anyone? Hello? Okay, so we'll tell like population or sample size. Population is like a bunch of or full data. Let's say in India we have 100 crore of data, so it that will become that population. Okay, but to test COVID vaccine or maybe sample, so we need to take the sample. So we'll take out of 100 crore, we'll take 100 sample, or 250 sample, or 1000 sample. So it's like it's something that we are going to take some sort of some small number from the population, so that will become sample size. Then we have to plan what is the purpose, or what is the objective. Then we'll do the data analysis, and then we're going to write the things and then we give the some protocol, and so that is going to follow from the follow that will go implemented throughout the country. What is the use of that is the biostatistic? So first one is to, so we are going to check the difference between the that is population, whether it's going to happen due to the by chance or by occurrence. It's something like we have to check whether some diseases are there, something is there. So we have to check whether there is a difference between the two populations is there or not. Let's say we have a we have to check there some immune property, immune system for male or female. So whether we have to check it out, like what is the things going on, like what is the difference between whether there is a difference is there or not, whether what is if there is a difference, what is the numbers difference, or we can check the correlation between the population attribute in the same population. If that let's say for a male, if 10 to 30 year or 30 to 60 year or maybe 60 to, like whenever that is the senior, that is citizen we have. So what is the correlation between the same population but the different attributes also? Either we can check the with the use of the biostatistic, we can check the efficacy of the vaccines also. Okay, so the similar way I have explained, that thing that need to be done, and we can tell what is the percentage efficacy, or we have to reject the hypothesis or something that we have to tell, or we can check the what is the measure of that mortality and then morbidity. What is the morbidity? Anyone? Morbidity, this is a mortality, something like what is the rate the people are dying for some sort of like some diseases and some flu. What is the morbidity? Anyone? Morbidity is something like we'll check with the what is the hospital, that hospitality rate, or what is severity, or we can check. Okay, or that the next thing will be to evaluate the achievement of the public health program. Public health program, let's say for a certain that age of the people, or something that some that municipal body they told, like they have to launch the program. Let's say that we are going to open a park, and we are calling that those who are 60 to 60 that more age. Okay, so 60 or that age we are going to call them, and then we have the we have the person they are going to monitor, and they're going to help in the walking, or some exercise, some yoga, and something like some exercise that need to be done for the senior citizen people. So what is the that health program was, whether it was that effective or not, whether it become good for the older people or not. So we can check the evaluate the achievement of the public health program also. So that program can be anything. Let's say they are going to run the polio scheme and polio vaccine scheme, or they are going to give some vaccine, or maybe they are going to ask do something the survey about something. So then after, based upon that, we can fix the priority in the public health program also. So once we'll check the evaluation of that some program, so we can give the some protocol, or either we can give some sort of something, so that is going to give their priority. After that, that is going to help for the promote the health legislation, and then create the administrative standard for the oral health also. So this is the something that people are doing, and then that is going to help in through the biostatistic itself. So that how a biostatistic can help, like let's say some people have studied the biomedical engineer, so they have the idea, and now they want to check, or they want to analyze the something. So they can take the help, or either they can learn by themselves, or they will going to take the help from the any biostatistician or biomedical engineer. So they are going to help into design of the study. Like, okay, first we have to tell the objective, what are the things I want, whether something that need to be done. So if you don't design the study properly, what will happen? Like we'll take the data in the that is the random way, or the data can be the noisy or something, so that not going to help you out. Then they're going to help you in the sample size and then power calculation. Sample size, someone might be knowing that we have the G power software to calculate the sample number or sample size. Okay, so in that we have to give, okay, what is the our and that object like p value, like whether we have to keep that is the 0.1 or 0.5, or like we have to that design like that objective like a beta value and all we have to give it. So based upon that, they're going to tell, okay, this much sample we need. So sometimes they'll say 12 or 50 or 500 based upon the power and all we have to do. So that is done by the biostatistician itself. And then after, if it does, what is that questionnaire? Let's say if that we have designed the things, we have calculated the number of sample size also. So then after, those who came for their experiment or the study taking, so they will asking some basic question, whether you were that you were having the injury in the last 6 months, or whether you were involved in some similar type of study in some other cases and all. Then after, the data management, so we have to check with their ethical parameters also, like how the sensitive data can be recorded or stored and all. Then after, based upon the objective and all, we have to choose the descriptive statistic also. Sometimes we'll choose the mean also, the mode, median, or maybe the skewness and all something, and then we're going to project into the graph. Okay, then after, based upon this values and all, we are going to apply whether that is a univariate or multivariate. When we have the one variable, so we will go with the univariate analysis. If we have the multiple variable, or we are going to take the repeatedly some sample we are going to collect, so we are going to apply the multivariate statistical analysis. Okay, so if we talk about the data collection, so data collection first will be that what is the steps? First step is to data collection. Once the data is collected, so data we are going to present maybe in form of the graphs, or maybe the histogram, or maybe the projection in the 3D on something. After that, once we'll see with the naked eye, but based upon this data, we can't say anything whether that data is we can't anything whether the data is good or bad, or the objective of the study, whether that we are going to check the efficacy, so whether that will affect it. So based upon that, we can't say. So now we have to come up with the, because this is the data presentation, we are going to draw some graphs and all. Then descriptive study. So descriptive study, so we're going to check what is the number, what is the mean, then what is the standard deviation, what is the that is the mean square error and all. So we'll check whether the things are clear or not. So now we have to check the what is the inference we'll get from this data. So then we have to apply some sort of testing. Maybe the hypothesis testing will be there. That we have to formulate. If we talk about the hypothesis, so we have the first is null hypothesis. Then we have the alternative hypothesis that need to be designed. Then after, we are going to apply some test, like maybe the t-test, maybe the ANOVA, maybe that chi-square test, or some that is non-parametric test also. Okay, so it's something that flow of full study, we can say. After that, if we talk about the that is statistics, so we have the two types of statistic is there. First one we have a descriptive study. It's something we are going to method for organizing or summarizing the data and presenting in an informative way. But from here, we can't get the conclusion. We can't interpret. Okay. But if we talk about the inferential statistic, the method that we are going to determine the something about the population based on the sample. We had the population. From population, we have taken a sample. Okay. Then okay. So we have a sample Then after we are going to tell then okay, so based on the sample we are going to tell something about the that is the what means about a population based on a sample. So in a descriptive, what they will do, they are going to just organize the data or summarize the data or present in a some informative way, but we can't interpret or we can't comment on the data type or the nature of the data or maybe something about their quality of the data. But in the inferential statistic, what we can do, we have to tell something about the population based on a sample, maybe due to the whether the efficacy of drug or maybe the treatment or something. So if we talk about the population, population is the entire set of individual or object that is our interest. But what happens when we will do the statistic and all, so we can't collect the data from all sample from, so we can't collect the like data from all population, because we have the 100 crores that 50 crores of something. So based upon the sample, so we'll take the sample, a portion or part of the population of interest. So based upon that we are going to tell. Analysis is performed on the sample to infer about the outcome at a population level. So that's why when we'll talk about the statistic, we'll never say 100% claim something, will not say 100%, will say our 90% confidence interval, like something is there, like that is going to be effective way. We will not say it's a perfect or something. So there is always chance of getting wrong also. So in a statistics, some statistic they will say okay, that degree that the drug is that much efficient or efficiency, but they will say 94% chances are there, or maybe their p value was defined like this only. So that's why we'll never say okay that claim is made like it's a perfect claim and all. Okay, so that statistic. So when we talk about the data, so we have the data, different types of data are there. First is primary data, second one is the secondary data. Okay, something primary data is something, you know what is that, like something that collected by the researcher or the person itself. So that data will say primary data. Secondary data, that is already available in a public domain. Those who are working in AI or ML, so they'll be getting data from the Kaggle. Or those who are working in the biomedical, so they have the num fees or some data. You will get the data set that is available in the public domain. But the data, like primary data, will be the always what you can say effective, trustworthy, but it's going to be expensive or time consuming. Okay, but if we talk about the
Excuse me, sir.
yeah, yeah,
Sorry for interrupting in the middle. The data collected from by the AI and ML, what is the Kaggle? Can you please explain it?
this Kaggle is actually a public domain website. There you can get a data already collected from someone that kept in the public domain, you can use for your uh, analysis.
So,
Okay,
what is the difference between this Kaggle and this the data collected by the biomedical these people?
actually the thing is that when we have the data, so one will be the primary data, second is secondary data. Secondary data is something someone have collected and that kept in the public domain. So that data can be accessed by anyone. Okay, but if we talk about the primary, that have been done by the any that is a research group or maybe the researcher, and they are doing the analysis. They are going to publish. So that data is something they have done by themselves. If Okay, they have done some analysis, so that is going to more effective, or more effective means something like that is most trustworthy. But the thing is that it will take time consumption, expensive data is there. Secondary is something that has someone already done. That let's say that we have the federal agencies are there, like ICMR is there. That is the Indian Council of, that is the medical research. They will collect some survey. They will kept in the public domain. If you want to analyze for that particular some survey, so that data you will take from the website and then do the analysis, and that you will come up with some sort of interpretation, something that you will come up. So that is something that has been done, but you are just analyzing it. Primary data is something that I have collected by myself, and I am going to analyze, and I am going to publish. So that's going to a different. So there are different types of data only. Okay, is it clear?
Okay, sir. Thank you, sir. Yes, sir.
So it's something. So sometimes, like we have a demerit and the merit for the both data. Primary data, I told it's effective, trustworthy, and but expensive and time consuming, because something before the collection of data we have to take we have to design, we have to the design of experiment, we have to that the monitor and all. But secondary data, it has been done by someone, some government agency and something. So that has been like they have already done, so they have the very good protocol. So sometimes data will be very good, data will be not in that, what do you say, uh, noisy and all. But if someone collected by himself, there will be chances of the getting data getting the noisy, and this data will be the publicly available, that you can use directly for the your analysis and all. So this is the some, what we can say, data. So this is the all about the data we have talked about. So now we'll come to the, we have the, once we have the data, so different types of parameter will be there. What is the constant actually? Constant, something that will not vary. Okay, can you tell someone example what will be the constant example in a, that the real life constant? Constant that does not vary. Let's say I have bought the AC. Okay, I have bought the AC. Now I want to install. Once you install it, so that power input is okay, that is also constant only. Why? Because the two, there are 220 50 hertz that will be given, or that is the, we have the one fixed that is the room size is there, that is also fixed. So that will become a constant. What will be the variable? Variable will be something that will change with the time, or maybe let's say some once the one people are there, two people are there, so it's going to be temperature will be like some 20 25. Once the people got increase, so what will be that we have to decrease the temperature. So that will become a variable, like characteristic that take different value for a different person, or the different, what do you say, attribute. So once we'll talk about the, for a different person, one thing can be the constant, so other can be the variable. But the other person, for them, it might be the variable, but the other can be the constant. Okay. For a designer perspective, let's say you bought some house. Now you want to make the houses. So once that architecture will come, so they're going to design. So for them, the plot size will be the fixed, so it will become a constant. So what will be variable? For them, the variable will be the room size, or maybe that may be the kitchen size. So there, for them, it's a variable. Okay, once design have been completed, so now the contractor will come to make the house. For them, there the kitchen size, or maybe their that the room size is constant. But for them, variable is, what is that? The quality of materials, then the time they will be taken, so that will be there. For them, it will the variable. So it's something like perspective of constant and variable, it will change with the person to person, is it? After that, we have the parameter and attributes. Can someone tell what is the difference between the parameter and attributes? It's already just written here, so just tell me what do you understand from here? Parameter. Okay, I'll tell you one example. Let's say everyone has the house.
Excuse me, sir.
Yeah. Tell me.
This parameter is that it describes like whatever the content which will be like population. Example here is that
Mm-hmm.
what the content is there in the attributes is that what is that exact
There, that is a that is a property. Property we can say, like a gender,
property.
caste, or
Yeah.
religion. So let's say we have
Yes.
a one example is there. Let's say we are going to buy the AC for a for a like buyer, or maybe we can say that once they're going to design it. So for them, it will the parameter will be something like wet bulb temperature, or they have to maintain the humidity and all. And if you talk about the attributes, attributes can be a 1.5 tons or for 2 tons, or maybe the different colors, or maybe the different sizes and all. So it's something they're going to take, they will give their properties, whatever the nature of the works or something. So they are going to tell the their about that is the population. So these things are basic thing actually that is going to help you once you will go into the deep of statistic or biostatistic. Okay. So now, based upon this all, so we're going to solve some question actually. Okay, we'll solve some question like, okay, so till now we have discussed about what is the statistic, why do we need to do the statistic, where the where that is going to use, and what type of data are there, what type of statistic are there. Some, then we have discussed some sort of variable. Can someone tell what is the difference between population and sample in the statistic that we have discussed already? Can someone tell? Population and sample, and tell about like along like with the example also. Okay, anyone?
So population will be our entire data, and
Okay,
sample will be like a small subset of that, which we can draw observation from.
yeah, like we are going to tell something about the population based on the sample only, is it? So it will become a entire
entire set of population.
the data set, or entire set of sample also we can say. Like it's going to be include all sample, and sample is something that we are going to take uh, some small number or a small number from population.
Subset of population.
Yeah, a small number or subset of the population. So let's say we have a, I want to check the COVID vaccine efficiency. Let's say they're going to check in a India. So for them, the population will become, let's say 140 crore. Is it? Now the sample, now the sample again, the taking the number of sample is always important. So simply we can't take by random sample. For the collecting of the sample also we have a different types of that the methods is there. Can anyone tell for collection of sample? Simply we can't say na. Someone will say I'll take 40, someone will say I'll take 10, or someone will say no I'll take 100 or 150. How come we'll come up with the one standard sample? Can anyone have the idea? Like anyone have the idea about this? Let's see. If I am going to check with the something that I have the company, pharma company is there. Okay, for that I have to check something. So what I will do, I will be taking some sample, but some other company will come. No, I will take this much sample and all. So how come a number that is going to be valid? Because some sample have taken some other company for their some sort of uh, inference is difference. For some company they have taken something, so their inference is different. Okay, so something will be there now based upon the something that we have to do. So what are the types of samples, or what are the method to do it? Anyone?
By
Uh,
use of power analysis,
Yeah, that is the one method that we are going to use the power analysis. So that will give the number of sample. Then we have the sampling, that method also there. Okay, one will be the that is the random sampling. Can anyone tell what is the random sampling? Random sampling.
Samples are selected randomly,
Okay.
2, 4, 6.
Okay, it's something we have a box is there, and a three red. That this is the fundamental example when we're going to start the proof. That is the, that is the probability and all. Three red ball, three blue ball, two green ball, something. So if you take the randomly, that can be of anything. So that will become your that is the random sampling. Second one, what is that? Systematic sampling. Can anyone tell what is the systematic sampling? Systematic sampling. Systematic sampling is something that we have to get into some order or some sort of set of rules. We don't have something go and take any things and all. Systematic name itself will say from this we have to take that much sample, from this set we have to take that much sample and all. So something that we have the rules, we can say. One more sampling that method we have a stratified. Anyone have that heard about this stratified random sample sampling? Anyone? Stratified sampling, it's again but like we have a some set of rules and something will be there, and or either based upon the observation or analysis, we have to come up with a take, we have to take the samplings and all. So this is the all about. So that's why we have to, whenever we'll say the samples should be a standard or that should be acceptable by the statistician. So we have to justify for this much of power, for this much of p value, or we have used this sampling method, we come up with this number of samples. So whenever any pharma company or drug company will say for a that much sample from the there is a population, so they have to explain these parameters also. Then they have to go to claim, okay, my drug efficiency or maybe the vaccine efficiency something 70% or 80% something will be there. Okay, so simply we can't say, because for a common man they will say no, I will be for me it's a convenience to take 30 people, I'll take 30 people. For someone they have very good manpower and all, they will take random, or maybe that 1,000 sample they will take and all. So that's why we have to be very clear or very specific about to choose the sample. Okay, so that is going to like a statistic, if we don't like do properly, it will tell a great lie. Great lie means we can say confidently, a statistician can say great lie. Why? They can take because a statistician is a human being. They can do the bias something, it's our human nature. Then we'll tell, okay, this I have took because and we have done some bias, and then we'll tell, okay, this data I have collected for this, but this much efficiency is there. So it's something, but we can say confidently because we are saying we are going to back with the some statistic value also. So that's why we have to be very broad, we have to be very open to choose the sample or the population or the sampling methods and all. Okay, so so population is the entire data set, or maybe the sample is the subset of the population. So again, choose the sampling also, we have the different types of methods are there. We have that is the one, we have the power analysis. In that also we have to, what is the method of the sampling also we have to think about that. So okay, so now we'll come to the second question. Can anyone tell What is the difference between the qualitative and quantitative data? Okay, just leave the data. What do you mean by qualitative? What do you mean by quantitative? Anyone, please? Anyone?
Qualitative data gives narrative form and quantitative data gives a numerical inferences. Something.
Yeah. Qualitative is something gives in the physical form, is it? Or maybe the appearance, or maybe the sense, is it? And the quantitative will be something that is given with the numbers.
Numbers.
So, can anyone tell what type of, what we can say, types of the qualitative data? Let's say I am saying like I have the pain, some muscle pain is there. So that pain will become a qualitative, is it? And same thing I'll take, I have the muscle pain of 5 board scale, is it? So this is become your qualitative, this will become your quantitative, is it? This is the one that is the, what I can say, scale is there, that the measurement style is there. So qualitative, that is something like something they are going to give the qualitative. Sometimes someone having a pain, that is the black color, the blue color, something. If someone said that 5 number of the 5, that is the black number, so 5 will become the quantitative. Or tell I have the 1 to tell that is a 1 to 10, we have the that is scale is there, on that I have the pain level of 7, so that 7 become the quantitative. It's something that we can going to represent on the numbers. So if we talk about this, all these data and all, so how can the measurement can be done for a data? Let's say we have the quantitative, we have the quantitative, and we have the qualitative, is it? What type, like what is the types of measurement, can anyone tell? Measurement type, either it can be done on the nominal scale, is it? Anyone?
Ordinal scale.
Then ordinal scale will be having, then we'll be having the interval scale will
The
be having, then we'll be having the that is the ratio scale will be there. Can someone tell what is the nominal scale? It's
is
something
focus on gender.
lowest measurement scale, shall we?
In case of pain scale assessment,
Yeah, that nominality means like something, it's going to start from 0 to 1, so this 0 will become like that, that is the nominal, but point 1, point 2, this all will become the ordinal scale. Point 0, 0 0.1, 0 0.2, 0 0.3, 0 0.4, 0 0.5, 0 0.6 to 1, is it? Ordinal scale is something like we are going to kept in the scale, and it's going to start from the -1 to 1. So this will become your ordinal scale. Okay. Then we have the third, interval scale.
is close to
Yeah, it's something that will tell in terms of the range, this much 5 range, 10 range or something. What is the ratio scale? Will tell low ratio or the high ratio. Based upon that we can something quantify, is it? So this type of measurement scale we have. So from that we can quantitatively we can measure in terms of this scales and all. So qualitative data is something like descriptive, blood type, diagnosis. Then quantitative we have the numerical. Then gender we have a qualitative, and then age is the quantitative. Okay, we'll come to the next question. We have a researcher. They are going to record the ECG signal. ECG, what is for? Can anyone tell? Chest, like this is that electrocardiograph actually.
Electrocardiography.
Yeah. For actually it's for that cardiac event to that the monitor. Okay. So that is a researcher. They're going to take the signal for the 100% and calculate the average heart rate and standard deviation. So just tell what type of statistic is this. So we have the statistic. Can anyone tell what is that? So we have a different types of statistic is there. Can someone name it? It's already there.
It's like descriptive and inferential.
Descriptive and inferential. I think it will be the only one only here.
Okay, you want to answer.
Predictive.
Oh, can someone please what is the inferential? Inferential statistic, what is that? Inferential statistic, something we are going to tell about the sample or population. Like we have to claim, we are something just claiming it, or something we are saying it, or we are telling about the something about the population or sample. Predictive is something like we are going to do some sort of regression or something, so that is a different thing. Descriptive, we are telling just a number or their physical attributes, maybe that the mean, but based upon that we can't say any something, we can't conclude. Okay, so then we have the casual. Casual is something, okay, we are telling this thing, but they don't have any inference, they don't have any description. Okay, or they can't do any predictive also. Okay, something will tell, okay, I have collected the data and I have this is the one value for this one value for this one, but they're not saying anything. Let's say they have done 100 participants collection, so they will tell a random in a casual way. Okay, for the one participant, the one the that the BP rate was something, that the beats per minute was something. So it's a casual way they told. So from here we can't inference also, we can't describe also, and even we can't predict also, is it? If we talk about the inferential, so we have to something claim about, okay, this this is more, that ECG device is having that much efficacy, they are going to that they have the range of this much, that much error will not come as something. So from this data, the thing they're saying average and standard, so we can't say based upon that thing, like we can't say. So now that is the predictive also, we don't have the setup also, so we can't predict also. Okay, so means here the average heart rate and standard deviation will give the descriptive value. Is it clear? So that third option is correct, is it? Or anyone have the doubt in this? Anyone?
No, sir.
Okay, so that is clear. It's something like, just see, whenever we are going to say or claim, I'll tell, okay, this much difference between the two participants, or this much difference is coming for this month. So then we'll say inference. Predictive is something, okay, now I have taken the data, I'm going to predict for the coming 100 population. So here we are not doing. Descriptive, it's something I'm giving, okay, I have collected the data for 100 participants. So for overall 100 participants, what was the value of the heart rate and what is the deviation between them, what's there. So it's a overall picture will give, so it's a descriptive value. Or here also casual way, I have taken the 100 participants, okay, one participant having that 1% having 100 that much value, second having this much. So we are saying casually for individual participants, okay. So descriptive, something they're going to summarize the data. It will going to tell us data set, but does not make a predictions or generalization. So this is the something we have to tell about that. So now we have a fourth question. Fourth question, we have a clinical trial tested a new pain gel on a 50%. Which statistic branch help to generalize the result to the all patient? I think I have told the description in the uh, last slide. It's something that generalize.
Descriptive.
Anyone?
Descriptive may be the correct answer.
Uh, no, it's a inferential. When we'll say about the descriptive, we'll talk about the number. Just see here, last slide. Descriptive statistics summarize the data. It's something the mean, the standard deviations and all. It will not tell us about the data, like it will tell about the data set, but not going to make predictions or generalization. Is it? So here, what is that? They are saying they have tested a new pen, that gel, but on the 50%. Now they are saying they want to generalize, or they want to make some conclusion, or they want to say something about the population. That is not done by the descriptive, because it's going to give only description. Let's say for this, let's say 1% was having, when the pain was there, let's say they were having 7, that is a rating scale. After applied gel, it become a 3. So this is for 1. For second, before it was 6, now it become 2. So if we talk about the before pain, 7, 6 plus 8 something by something, that number of that is the participants or patient, they will give some value, let's say 6.5. So this is the average value. So this average value described by descriptive, is it?
Yes.
And if we talk about, okay, this for this new pain relief gel, what is the efficacy or efficiency, like after applying the gel, how much percentage got reduced, or like maybe that from, let's say 6.5 rating scales to how much it came, let's say 2.5 or 3. So we are making the claim. So it's based upon the inferential statistic. So for this particular question, that we will be having the answer of inferential statistic. Can anyone tell the diagnostic statistic? What is that? Or experimental statistic? It's just a method. Don't confuse with that something. We'll be having only two types of statistic. One is descriptive. Second one is the inferential. In that starting itself, once we're going to present the data, we'll tell the some sort of, what we can say, overall data set. Will not make the conclusion, but inference will going to make some conclusion. Diagnostic is something will tell after the inference. So what is the diagnostic label? That, let's say we have make one machine like ECG, that the machine is there, whether 10, that 10 machine we have, they have done 100%, 100% have done. So now someone have done 80, their accuracy is 80%, 90% and all. So they will say out of 100%, 70 diagnostic level or maybe that much. So it's something like they are going to check or they are going to able to predict something. Just see in a medical, we can't go 80, 90. That should be 99%. That error, that margin of error should be very less. Is it? Once you talk about the, once you will say okay 80%, what will happen if that things are coming 20%, there is a chance of getting the person will get die na. So whenever we'll talk about the any medicine equipment, medical equipment, so we have to think about there is a margin of error should be very less. Either it can be a from the machine side or maybe that from the any doctor side or maybe the nurse side and all. So that margin of error should be very less. Experimental is something we are doing the experimental, we are checking it and then we are going to generalize. So it's experimental methods are there. So this question is clear na. When we will talk about the generalization of the population or sample, it becomes inference. But once we talk about the data set, it's what is the average, what is the mean, what is the mode. So it will say descriptive. Is it clear? Okay, we'll go to the next question. So if a 50% show 30% reduction in pain, inferential stats help us to estimate how the gel will perform in the broader population using the confidence interval or p-value. That's what I try to explain here. Okay, this slide I'll keep in the what I can say, like I'll be going to upload so that you can access actually. So that thing I will remove it and then I'll try to keep on the website, NPTEL, that is NPTEL website. Now we'll come to the next question. Next question is that which of these is an example of primary data in biomedical research? Can anyone tell? Published data set of SCMG signal. What is the EMG? Can anyone tell? It's a electromyo, where it use, it's a electromyography. Where is use myo means we have the muscles. We have the ECG also, no? So electrocardiography. Then we have the EEG also. Anyone have the idea? What is that?
Electroencephalography.
Yeah. Something is there. Graphy. Then we have the MMG. I will explain this thing also. Like we'll go a bit slow. Like if someone having a broader area of the their that what you can say research. This is all the medical equipment that is be used in the biomedical. So we have the MMG, mechanomyography. Any other device? We have non-infrared MS. That is the muscle scan. Then we have a invasive EMG, like needle type. Any other thing? CT scan is there. Actually, now those who are studying the EMG, they are using that NIMS also and CT scan also for their research part. So electromyography actually deal with the muscles. When we're going to do any activity or exercise, what will happen? Muscles will get contract or expand. From this contraction and expansion will get a electrical activity will get it. Okay, electrical activity. Then it will become a signal. Then we can analyze it and then we'll tell what is the conditioning of the muscles will tell. So this is all about the electromyography. Electrocardiography, they are going to tell about the heart rate or maybe the BP rate also. Then we have the QRS value also. What is the QRS value? Anyone tell? Yesterday only I had the ECG actually. They were having, but all the normal, no issue. So it's something that going to deal about the cardiac or cardiology. Those are the cardiology. They're going to check about the there when the cardiac arrest happen. So what is that? What happened with the heart rate and all. Electrography is something that going to deal with about the brain. Is it? Those who are doing the work in that they are going to brain interface or maybe the cognitive function. Okay, mmg is something that we are going to use IMU sensor, or it's going to measure what, like vibration. If we keep on the muscles, so they're going to keep the muscles vibration. Let's say whenever we are going to that run and all, after running what will happen, that muscles will get vibrate. Just see, with the naked eye or we can't sense or feel, but the thing at the very, that is the lowest scale level, that is the similar case of EMG also. What happened when it's going to contract and expand, there is that is the electricity is going to generate, but we can't feel, we can't, if we touch also we can't feel, but the thing is that that sensor will take care. So there, that frequency is very low. So but the thing is that based upon this data, muscles, they electrical generated, because they will tell the motor unit activity or the muscle activation, whether if a trained participants are there, their muscle activation is more. So it means if they are going to more superior, they are not going to fatigued out. So it's going to be like they will not get the injury also. So that thing we can check with the EMG also. Similar way also, if the muscles vibration is more, what will happen? The vibration is more, there will be entropy will be more. So they will be, they're going to drain out. Then they're going to, energy is going to deplete. It's going to be like again the injury will comes into the picture. So it's all about the biomedical devices. Then we have the EEG. EEG is going to, let's say I am going to do some functions. Let's say I am writing or I am doing some activity. What's happening? How the vein is connected with that particular muscles. Let's say doing the writing, I am going to keep the EMG on the forearm. Okay, so during that particular the forearm EMG I have calculated, but that is the forearm is connected with the, that is the brain. So we are going to keep the EEG here, brain. Then we can going to map between the muscles and the brain also. Okay. Similar way, we have the NIMS. It's going to be a non-infrared. So they're going to check the muscles conditioning, whether the muscle got tired, muscle got teared out or not. Something. The CT scan, we can check the area of muscles. I am telling the context of the some sort of that is the research. Okay, I think it got bit of more. I will come back to the question. Okay, tell me which of this example is the primary data? Can anyone tell what is the primary data and what is secondary data? I have explained already now.
She directly collected by the researcher to the subjects.
Okay. So first option is the published data set of the SMC. This data in which category? Secondary. It means someone have already collected, but it's published in a public domain. Is it? So this, so this one, this one become a secondary data. Third one, we have a lab manual with average stride length. What is there? Lab manual with the average manual stride length.
Secondary data.
It becomes secondary data. How?
Because already it is there manual.
It's already someone have took, someone have already done the analysis. They have given that is the description for this type of gate walk 1, this much description average is there. Gate type 2, can anyone tell what is the gate cycle or gate? GAIT. Okay, well, gate cycle is something that the people are walking, running something. So if doing the whole stride and then what we can say, that swing phase and all, that will say the stride length, like gait cycle, one gait cycle. Then we have the fourth one is clinical textbook value of FEVP1. It's something medical term that I also don't know, but clinical textbook they are saying, so it means something they have done, clinical value is there, so this also will become a secondary data. Okay, then we have a real-time data collected from the volunteer using a chest strap. It means something that a person is doing currently. So it will become a primary data. Is it clear?
Yes.
Yes, sir.
So primary data is what you collect using sensors or devices. Secondary data is already analyzed or published. Knowing the source is crucial for the reproducibility. Okay, so this is just a description I kept. Okay, we'll come to the next question. We have a which are the qualitative variables below. It's again something is there. Can someone tell what types like types of the variable? Anyone able to answer the question?
Posture,
Okay,
gender,
okay. Any other? As we all know that we'll have once we talk about the variable will be having two types: qualitative then quantitative. Is it?
sir.
And if we talk about the variables also, we can have the random variable also. Any example?
And a variable, we take as a first row, third row, fourth row.
Yeah. Then we'll be having the discrete random variable. In the random also we'll have the different types: discrete, then continuous. Okay. Discrete is something characterized with the some gaps. Maybe that we'll be having the interval, or this continuous can become a maybe the signal. Is it? So once we talk about the EMG amplitude it's a something. We have the number. Okay, let's say we have a this the curve is there. So this part will become a amplitude. Is it? That amplitude having some number, maybe the 10 to the power 1 second, if it is this volt or time, so this will become a volt. So this is all the numbers. So this is the quantitative variable. Is it? If we talk about the point level, it's something of a scale: 1, 2, 3, 4, 5, 6, 7, 8. 1, this is 2, 3, 4, 5, 6, 7, 8, 9, 10 is something they're telling about the numbers. So it's again will become a quantitative. What about the gender? Is it qualitative or quantitative?
Both.
It can be a both. But once you talk about the posture, sitting, it's a qualitative. It's something a person can be like it's a physical things attribute and all. Standing, it's again a something like qualitative. Lifting, it's again a qualitative. Then again we'll say sitting at which angle. Then that will become a quantitative. Is it? Let's say for a car seat and all will tell okay that much percentage of that degree and all something should be there. Standing also they will tell like, okay, standing, it's something that we can't change the variables and all. Lifting will tell, okay, this much weight is they are going to lift it, or maybe like that is the lifting posture is that much. So something that will become quantitative. So here effective answer is second, that force can be both, you can say. First one is the quantitative, second one is qualitative. This is also quantitative ordinal, but qualitative, it's again comes under the both, but it's, if we talk more into that, it becomes a qualitative nominal scale. Nominal scale. Is it clear? So so that's why whenever we are going to start the statistics, so we should be very clear what type of the data or nature of the data are there, whether it's a qualitative or quantitative, and based upon this only we can able to come up with any hypothesis. Simply we can't come like something if we don't know. Let's say someone will take quantitative as a gender also, or maybe the possession, all it's something, it's again, if it says setting 1, setting 2, setting 3, setting 4, they are different posture angle, so that will become quantitative, so that will become variable. Okay, but if it is nothing is given, so it's become qualitative. Let's say for the sprinter, they have to do some training. For the training they will do some exercises. Exercise is become a qualitative. In the exercise also we have a different types of exercise is there, so that's also qualitative, but on the uh, nominal scale. Let's say for a strengthening of the lower extremity, we have a squat, lunges, we have a different that is a leg curl, leg extension, so it's again qualitative. But someone say squat they are doing at 50 kg, 40 kg, 30 kg and 40 kg. This is the quantitative data. Okay, someone told the lunges, it's a qualitative, but they are doing at the different weight level, so that will become different weight level, so it will become the quantitative. So that's why I'm saying, like, before jumping into the any of the statistic or the future, if that course you have taken for the foundation for the machine learning and all, we should be very clear what type of data is there, what type of variables are there, then we can go ahead or we can go forward. Okay, is it clear? Yeah. Now we'll go to the next question. Next question is that we are going to uh, analyze the data from a blood glucose monitor used in a diabetic patient. Okay, we are going to analyze data from blood glucose monitor in a diabetic patient. What type of variable is blood glucose level? Can anyone tell? I will explain actually, but try to understand like what type of variable is there: categorical, quantitative, or quantitative also discrete, or quantitative continuous, or maybe the ordinal variable. Please tell.
Quantitative, discrete,
Can can you please tell the some example of the categorical variable? Categorical variable is something class A, that can be of anyone, the boys, gun, girl. Class B again. So it's something that we are going to categorize. Is it clear? Or let's say for a training purpose, we are going to plan training A, training B, training C. In that different types of categories are there. So this will become categorical variable. Okay. So this will, that the blood glucose level, it's not going to become a categorical. Is it okay? Now we'll come to the ordinal scale. Anyone? We have that discussed in the last question also, like before last question also. Ordinal scale. Tell in the context of blood, that is the glucose level. Anyone? Just say, ordinarily something's scale is there. Let's say pain level, that we are going to put on the 1 to 10. 0, this is 10. So for a glucose blood level, that we don't have this level now. For a person, one person can be about 250 or 150, 100, something we don't have the limits, so that will not become an ordinary scale. Okay, now we'll come to the quantitative continuous. So now we know that it's a quantitative, but it's a continuous. Whether it will be the continuous or not, tell me. Continuous or not? Anyone?
No, sir, you explain.
Okay. Can you tell the definition of continuous? Let's say we have the time, it should vary like this. So it's something, but when we'll say the glucose that is the blood level or something, we'll tell, so we'll tell in the directly, okay, this much value is there, this much. Okay, during the monitoring with the time passes, so that will become, the signal will become continuous. But when we have the any monitor, they will give 80, 120 for the BP. So this will become quantitative but discrete. Is it? So it means like for something BP monitor, glucose level, during the monitoring, once the time passes, so signal will come, because of they're going to measure the heart rate level for the BP. For the glucose, what they will do, they're going to take the sample of the blood from that pinch of the blood from the finger. Then they're going to put in a machine, paste upon they will give a some value, one single value they will get, maybe the 90, 100, 250 and all. So it's something we don't have the, what we can say, continuous, is it? So that will not become a continuous. Okay, so it's a discrete. Can you tell some example of the continuous data? I told when we are going to measure the ECG, we can see with the each second will give some value, or maybe the EMG, it's also giving the with the respect time. So it's a quantitative continuous data. So means for this question, once we get the blood glucose level, so it's going to be a quantitative discrete value, not the continuous value. Is it clear, this one? Is it clear, or any doubt is there in this? Okay, we'll go ahead next. So glucose vary along a real number line. It's a continuous, but it determines which kind of plots or test you are going to use. So it's something, whether they're going to take the value, it's a continuous. But the thing is that once you're going to give a last value or something, so it's going to give a number only, not the, what we can say, it's not the continuous, because it will give a single value. When we're going to glucose meter, it will give a, once we will pinch the blood, it's going to give single value. But process during the time, it can be the continuous, the similar way of the ECG or EMG also. Okay, so this is the, for this we have the correct option is the quantitative discrete. Now we'll come to the next question. Which of the following is a qualitative variable in a vaccine study? So our keyword is that qualitative variable. What is the definition of the qualitative? Something physical attribute, physically or attributes. Can anyone tell, like, let's start from the first one, what can be? time to immune response. Time to immune response is something they are going to give a some timeline. When will say like when you're going to apply the vaccine or giving the vaccine. So what is the expected time to immune? Just 1 minute. Okay, so they will say what is the expected time to immune. Let's say if someone having someone, let's say one population having some TB drugs, TB. For that, they're going to administer some drugs. They will tell, yeah, that we have to administer for the 6 months. After that, it will take 2 to 3 months. It will going to cure out. So they have given some timeline. So it can't be the qualitative. It's a quantitative. It's not the qualitative. It's a quantitative. So it become a quantitative. Okay. What is the age of the patient? What will be there? Age means again a number. Okay, whether it's a it's giving a number. Age of the patient, it's something. Let's say they will take population age. They will give uh, like average age or standard deviation something, or they will see the sample. They will give the again average standard deviation. So it's something a quantity. Okay, it's not again, don't confuse with the descriptive and inferential. We're not talking about the descriptive. So descriptive, it's again a average, that age of the person, it's a description. It's average, standard deviation is a description, but we are talking about the variable type, qualitative variable or the quantitative variable. So it's a number. So we have to just think about the whether it's a physical or attribute something. So it's become a qualitative, or it's a age of person, it's a number, the quantitative. Whether or not side effect occur or not, it's something we are telling that qualitatively. It means we are giving the something, the sense or like effect, but we are not giving the number, is it? We are not giving the numbers. So it's become a what? It became a qualitative variables. Okay, in this also, what are the percentage of the side effect? Let's say I have administered this particular drugs for 100 people. Out of that, 5% of the people got side effect. So means that 5% will become a quantitative variable. But now they are saying whether or not. It's something I am just qualitatively I am saying. Okay, whether its side effect is or not. That's all. So it means a qualitatively I am saying. I am not saying based upon the number. So for this question, we have the answer whether or not side effect occurred or not. So the fourth, what is that? Number of hospitalization. Let's say that vaccine, the COVID happened, COVID 2020. In starting the February, then January time, it was saying it's under the China only, nothing will happen, nothing will happen. Once it started, it's become a some sort of what we can say, like it got like too much number, and the hospitalization rate it increases when the time passes. We that the COVID variants came, that the Delta variants came, Omicron variant came. Okay, so for that particular case, for that particular what is number of hospitalization? So in that particular case, what you can say, it's telling about the what was the number. So it will become a quantitative. Okay, so for this question we have the answer of, so When we say not everything is numeric, okay, a simple yes no about side effect is a categorical or often analyzed with the chi-square test. Okay, this test will see the further classes and all. Okay, so whether this question is clear now?
Yes, sir.
Okay, we'll go next question. Next question is that we have the going to analyze the SpO2 data. What is that actually? SpO2. It's something we are going to make oximeter. It's again will give something the heart rate and all, or actually the oxygen
Oxygen saturation.
oxygen saturation actually. Sorry for the heart rate. Actually, heart rate will cardiac will give. It's the oxygen saturation will give. Okay, so we have the that is the oximeter. They have made they have used during the sleep in patient with the sleep apnea. Some sort of that is a disease is there. So what type of variation would a statistic help to quantify? Can anyone tell? It's something that a statistic. What is the definition of a statistic? Collection of data, then analyzing the data and tell something about. So collection we have done already with using an oximeter. Okay, so collection done. Now we have to tell based upon their parameters or something. So total that lung capacity, it's something totally, it's going to be uh, irrelevant with respect to the this particular example, is it? We can't comment based upon the oxygen saturation levels and all. We need some more sophisticated, okay, pulse oximeter that can give that is a overall picture, but will not give the quantification or something they will not give. So total lung capacity from this we can't get it. So it's something that is going to be totally irrelevant to the particular this one. Okay, so this can't be. Second one is the mean and standard deviation of the SpO2 dips. So something they will tell, let's say some person was data collected. So let's say for each second or for something some data came. Let's say like 98, 97, 96, sometime 86 also went, 80 also went, or something 99 also went. Okay, so now we have to check, okay, mean, or based on the division value we can tell, okay, for overall picture what was the overall. It's just 97.5 plus minus. Maybe if the variation was more, so it become 10. If it is variation was less, so it will become 5. So based upon that, so a statistic will tell. It's again I'm not saying which statistic. A statistic can be of descriptive, that deal with the overall picture like average, standard deviation or something. Inferential, something commenting about the population based upon the descriptive study. So here, so mean standard deviation can be a statistic to quantify, is it? That variation can be quantified with the help of this mean and standard. What about the CT image segmentation? It's again totally irrelevant for this particular example. But this again CT image segmentation, again if we go with the different context, let's say like I am doing the exercise and I have done the leg extension. Leg extension, like some people that might be doing that sitting on the posture, and like they will do, and then there is a weight is there, and this is the leg. They're going to apply the force. So they're going to target the quadriceps muscles. Let this is the person. They're going to target. They would target the quadriceps muscles. Okay, I have done with the 50, 60, 70, 80 percentage of MVC. MVC is something that I have, let's say my capacity to lift 100 kg of weight. So percentage of there. So I have done with the 50 kg, 60 kg, 70 kg and 80 kg. So now after each of exercise, each 50 kg exercise, I have done the CT image, and now I am checking with the what's happened with the muscles fiber. Okay, then 60% what happened with the muscle fiber, 60%, 70% what happened with the muscle fiber. Now we'll check what is the segmentation changes came. So if that analysis has been done, so that is okay. Like it's again becoming that is the inferential statistic will tell muscles that much got tired, muscles area that much got changed and all. So it's again a different analysis. But for this particular, what we can say, sleep with the SpO2 or that is a oximeter, it's a totally different parameters is there. Okay. So for this we have a different application. If that context will come, so again it's going to be a make some sense in that particular sense only. So I think I don't know how many are you are and that PhD level or not. If in the BTech level, it's going to help you out somewhere, like which, because it's a biomedical course that you are going to study for the biomedical devices. So what is the purpose of the biomedical devices? Is that all that you will see out. If someone is doing the research or PhD and all, so if they are into the that is the muscle physiology and all, it's going to be help more. So for all broader picture, maybe that biomedical engineer, that is the researcher, that PG course they're doing, so that statistic everywhere is going to help you out. And this statistic actually a foundation for the machine learning course. So if you want to learn the machine learning, you should be having very good idea about the statistic. Along with the statistic, you should be very good in the what you can say matrix or the maths also, algebra. Okay. What is the fourth option? Oxygen concentration in the hospital room. With the one patient or an all, we can't take the concentration of the hospital room. For that, we'll be having some different devices will be there. Or either if we say, no, like I have 110 patients are there. I kept in the in the room, and I am going to take all the participants, and then I'll going to give the that will give the overall picture of the room. That is not correct, because that might be the different way. If the some person is using the AC, if that in particular room that AC is there, so it's giving different something. But their SpO2 will be that will be different. So we can't generalize. So for the oximeter, this context will be wrong. This also not correct. This also not correct. So that is a correct answer. That correct answer for this. We have a mean plus standard deviation of SpO2. Is it correct?
Yes, sir.
Okay. We'll go to the last question. So biomedical engineer often need to quantify physiological variations like how often SpO2 dip below 90%. If it is going to below to the 90%, it means something abnormal things can happen. So this is the more part of doctors and all will see. As a statistician, we'll check, okay, oh this much down went, okay, that should not happen. We have to go and tell it to the doctors and all. So that will comes in the picture. Next question and last question we have a, which of the following best illustrate the use of descriptive statistic in a hospital setting? It's something we have the keyword descriptive. Descriptive is that we are not going to take any interpretation or will not claim anything, will not do this thing. We are going to just tell what is the something will be there. Okay. So what is the first example actually? Estimating the survival rates in the cancer across all of India. What is that actually? Tell me. How can you tell? It's again a estimation. It's again a estimation of survival rates. Let's say a cancer vaccine was there, or not cancer vaccine, it's something in India 1 lakh cancer patient is there, their treatment is going on. So what is the survival rate? Survival rates, it's not I am saying 100,000 is there, that much average their tumor level something, and then what I am not saying, it's not I am saying description or descriptive. What they're saying, survival rate. Let's say 1000, 1 lakh people was there, in that 800 died. or something 500 got died, something like this will tell. Then based upon that we are going to check, okay, this much percentage, like estimating survival rates, it's something we are going to tell or infer. So it's going to tell inference, so it will become a inferential statistic based upon the past record of data, based on past record data will tell like this much is the survival rates in cancer across all the India. So whenever any patient, new cancer patient will go, they will check, okay, out of 100, 2 people have died, or maybe the 10 people have died. So it's again, or 90% survival rates is there, or maybe the 10% that survival rates is there. It came from where? It came from the inferential statistic. Okay. Then after, so it that become, that will be the not option, because descriptive it should be a like something average or something we have to tell in terms of broader picture, not to tell survival rates and all. What about second one? Testing the vaccine efficacy. Again it will become a inferential statistic. Is it? How come? Let's say COVID came 2020, that starting 6 months, we 6 months, we can say 3 months that we were not having the vaccine, and it was spreading all over the world. So and now they have come up with some vaccine. Okay, and now the vaccine is that, so they can't expose, because just see, whenever the vaccine is there, so something vaccine is going to be an antidote. So something we have to make another virus that is going to be immune to that particular virus. Is it? So means, so when we are going to make the vaccine, so we can't expose to the all population. We have to take some samples. Some samples will take it, and we'll do the testing, and we'll do all the analysis and all. After that we'll say, okay, this much efficacy is there. If efficacy got 60 to 70% or 80%, okay, this much efficacy, they will come into the market, then pharmaceutical company will make it, and then will tell my vaccine is this much the superior. Let's say in India we were having Covishield, Covaxin, and then Sputnik was there, then that is the booster came. Who first told Covishield, okay, that my efficacy is somewhere 60-70%. Then the Covaxin came, no, mine that efficacy is this much. Then after Sputnik came, no, this much is there. Okay, they told, okay, this much is there, but now I am coming with the booster. So people took the booster also. Is it? So they are going to tell efficiency of the vaccine for that particular virus or something. So it's again a inferential statistic. The third one is the predicting the spread of infection. Let's say some flu happen every year. Okay. So let's say that some rain came, some due to that some flu came. So now based on the previous year data, the last year some flood came, due to that some flu happened, this much people have died, this much got infected, this much people got hospitalized and all. So based upon there, and what are the severity, what are the things was there, so it's all coming with the inferential statistic, and based upon that we are going to make the forecasting for the coming year, or maybe the prediction, or maybe the will apply going to what you can say, forecasting will say something about the future. It's again a what you can say, inferential statistic. Is it? So now third one we have a calculating the average ICU stay duration from the hospital records. We are not saying whether that ICU person, that let's say one person that patient came, whether what was that is the severity of the injury, what was the uh, nature of the disease and all. I'll not say, I'll not bother about. Only I am saying one participant came, that casualty ward, for them what was the average stay was there. It's a something description, nothing more about the claiming, nothing more about the something. So that will become the descriptive. Is it correct? So for this, we'll be having the answer of third answer. Is it correct? Anyone is there?
So,
Yes, sir.
So I think this is all about the clear, na? So any doubt is there?
no, sir,
No,
it
sir.
was clear today. Today's class was very clear.
Okay, thank you. So, like that, I will try to summarize like what I have discussed, because the week work and tech was the introduction of biostatistics. Like we have discussed, why do we need, what is the role of biostatistician, then application of biostatistic, then what type of data are there, some example we have took. Okay,
Types
and
of
the,
statistics.
yeah, types of statistic we have also discussed. We have discussed about the types of the statistic also. So this was the all about what we can say, like, so giving the brief of the statistic, how come it's going to help in the future. And the coming week we'll see that what is the descriptive statistic, what is the role of probability in the statistic, and we'll discuss about the what is the need of statistic also. Okay, and any can you just give some feedback like how the session today, I think one have given, or where I can improve myself.
Everything is clear, sir. And I have few doubts that is it this classes will be once in a week?
Sorry, I your voice is bit of breaking.
Now you can hear, sir.
Yeah, that little bit breaking. Okay, tell me.
All the topics are clear. There is no doubts. Also, you are clarifying the doubts. It's actually sir, very useful and it is very understandable, sir.
Okay.
And, yeah, do we have classes once a week, sir?
Sorry, like, can you just type it? I'll just try to
Oh,
get you.
okay, sir.
Huh. Any other? Can you please tell the feedback there so that I can improve for the class? And I'll do one thing, like I'll keep this slides and all in the website that you can access there.
Okay, okay, sir.
Okay, so we'll see again on the uh, next week Saturday for the second week type session.
Once in a week we'll have classes, sir.
Sorry.
Once in a week we'll have classes, sir.
Yeah. This will be that will be on every Saturday at 4 to 6 p.m.
Okay, sir. So, you will send me this PDF in this only, sir,
Actually,
for
I am
the
from
email.
NIT Trichy. I'll show my profile itself.
Okay, sir.
This is, and I'm working in some sort of a sports biomechanics and currently PhD scholar and PMRF scholar.
Okay, sir.
And if you have some doubt and all, that you can ask in the forum. I will try to reply there. And if you need
Hmm.
any other help and also just keep it there. I'll try to that reach out.
Okay, thank you, sir.
Okay, thank you. So shall we wind up the session? Shall we wind up the session?
Yes, sir. Sure.
Okay. Thank you.
Yes, thank you so much, sir. It was very easy, able to understand and grasp the material because everything was displayed there, so you are able to understand the questions as well as the explanation was very, very clear.
Okay, thank you. And that I will try to keep as the same, and I'll
To
try to.
revise once again.
Sorry.
So this content, as you said, you will put it in the online so that we also able to see once again and
Yeah,
see
sure,
the
sure.
explanation, whatever you have explained. So
Yeah,
thank you so much.
that will be kept on the that YouTube also. This video will be on the
Okay.
that YouTube also.
Okay, sir. Are there any books to refer,
Okay,
sir?
sir.
Sorry,
Are there any books to refer?
book. I can say one book is there. I will try to type.
Okay, sir.
Actually, the book name, that's a biostatistic. Okay, I'll type it. By uh, Bain W. Daniel. And the publication is actually that Wiley,
Okay, sir.
that you can refer the book. Actually, that book will be enough.
Okay, sir.
Okay, okay, okay. Thank you. That will see you in the next week.
No, okay, sir. Thank you.